Hi guys. Some days ago I went to hospital for corona test because I am falling ill for 14 days and have some acute respiratory syndromes. When I went to the doctor for test to the hospital, he collected some fluid from my nasal passage and put a drop of it in a spherical structure in a test kit. He said that I am corona negative. It is my fortune that I am negative. I asked him what it was. He replied that it is Rt-PCR test kit. Then some questions arise in my brain that what is PCR, what is RT, what is Rt-PCR, who discovered the PCR, how it occurs. After many findings, I am concluded that
Carey Mullis discovered PCR in 1985. PCR is a method that is used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally called Rt-PCR. Virus is typically present in the airways of people infected with COVID-19. A sample from the mucosa of the airways can therefore be taken and used for PCR analysis. Then RNA is purified from the sample. When RNA has been purified from the sample, the first step is to change it into DNA. This is called reverse transcription and it is done with an enzyme named reverse transcriptase. By this, a complementary strain of DNA, cDNA, is created, a kind of mirrored copy of the original strain of RNA. The enzyme Taq polymerase is then used to create another strain of DNA so that double helix DNA is formed. Then the PCR reaction starts. It is a series of temperature cycles that generates a lots of copies of the selected virus genes. Specific primers are used to ensure that the only selected virus genes are amplified. Then denaturation occurs, annealing occurs, and elongation takes place. This process is then repeated many times, ending up with a lot of copies of the selected genes. For detection, probes labbed with fluorescence can be used. This will tell COVID-19 in the sample.